Good morning. My name is John Alkan, and this is how you pronounce my name. It is John. It's not Ken. It's not Khan. Please learn how to read people. So the last talk of the day will be from Cenk Sahinalp, but he's not here. It is Cenk. All right. So you already listened to some talks about structural variation, but you are going to have the tutorial on the last day. So now you can learn what this structural variation is. The genetic variation comes in different sizes. The most widely known, the most as the number goes, are the single nucleotide polymorphisms, which are the changes of one character. By definition, up to 50 base pairs of changes are called small insertions or deletions, or indels in short. Or that could be microsatellite expansions and contractions. So the larger ones, more than 50 base pairs, are called structural variants. So this could be deletions, insertions of novel sequence, or non-reference sequence, as we have listed in one of the affinity talks, or mobile element insertions. That could also be segmental duplications. So these are typically defined as duplications of size larger than 1 kb, but obviously there are smaller ones. There are also inversions of DNA and translocations of DNA from one chromosome into the other one. And the much larger ones are called chromosomal changes. The most typical example is the 21 trisomy, which causes the Down syndrome. So these are the visualizations of the same variants. We have some deletion and some deletion of DNA. If it is an important gene, it could cause disease. Diseases such as autism, developmental delay, or Crohn's disease have been associated with various deletions. There could be new sequence insertion, so this is a sequence that is not represented in the reference genome. There could be copy-paste events, which we call the mobile element insertions. So these already exist in the human reference, but they can copy and paste themselves. So for example, the ALU sequence is about 300 base pairs long, and each human has about 1 million copies of it. And only a handful of them still can copy-paste themselves. And hemophilia has been associated with an ALU insertion. There could be tandem or interspersed duplications. So far, by definition, they have been 1 kb minimum. But again, you can now find smaller events as well. And schizophrenia and psoriasis have been associated with these. There could be inversions of DNA, so we just flip the DNA, parts of the DNA when compared with the reference, or we can copy and paste. We can cut and paste from one chromosome into another, which is a cause of leukemia. So these are the copy number variants because they change the amount of the DNA in the cells, and these are called balanced rearrangements because they typically do not change the amount of DNA in the cells. So why do we study structural variation? One, it is common, although we can expect about 3.5 million SNPs per person on the average. We usually see around 20,000 structural variation on the average in a human genome, but they can be everywhere, although there are some hotspots. So this slide is pretty old, from 2008, where Jeff Kidd constructed a human genome structural variation map the first time, using 9 human genomes using the plasmid and sequencing, which is the first sequencing technology used for this purpose. So as you can see, there are hot spots in the genome. So they are rare, 20,000 per genome, but they are collectively common. So they could be anywhere in the genome. And they actually affect more than 20 million base pairs per human. So the SNPs affect 3.5 million total on the average per human, and structural variants affect 20 million. So they also cause disease. So it can cause disease obviously in multiple ways. So for example, let's say we have a duplication of sequence. So those green bars are duplications, and those duplications may misalign during meiosis. And because of this misalignment, you can have an extra copy in one gamete of the green sequence, and not only the green one, but the ABC in between, right? That was unique to start with. And in the other one, you are missing that ABC and part of the green sequence. So again, if it hits an important gene, it could cause disease. So for example, 1% of mental retardation in European populations have been associated with a deletion in chromosome 17. There are also multi-copy genes, so these are the segmentally duplicated genes. So each chromosome has 2 copies, one comes from mother, one comes from father. But because of those duplications, a gene can be 3 copies, 5 copies, 10 copies, right? Increase and decrease in copy number of some genes are also associated with disease. So for example, the defensin genes, the DEFB4 gene increase is associated with the psoriasis, and the decrease is associated with Crohn's disease. I'm guessing all of you know how the sequencing works. So basically, I'm going to deal with sequencing in 3 different technologies. So we have the test genome, we randomly shear it, and then we do some size selection depending on the technology that we use. Or then we either sequence in paired ends using Illumina, or we sequence longer. We generate longer reads using PacBio or Nanopore, or we can sequence in shallow depth using linked-read sequencing that I'm going to call as long-range sequencing. The typical example is 10x genomics. So just a quick example. So we have the sample. We don't actually know the exact order in the sample, but we just generate randomly short strings from this sample, and we oversample it. And then we compare it with the reference genome. With long reads, these are much longer, but the error rates increase. So those green, those red fonts are errors generated by the sequencing machine. And then we have the linked reads. So these linked reads are actually use Illumina data. So it is the same short reads. They are more reliable short reads, but we can also link them together, saying that they are within 50 kb of each other. So the top red box, for example, we have 2 short reads, but we know that they are coming from the same large molecule, which is on the average about 50 kb. So these are the sequencing technologies. The short read Illumina, we have short reads, but they are also paired, and that means from one 500 base pair molecule, we have a prefix and a suffix. We have billions of them. The error rate is very low. The long read platforms are the PacBio and Nanopore, so we can generate up to a mega base pairs. You already listened to Mike Schatz's talk about how the long read technologies work. They are not paired and they are single, and we still have millions, hundreds of millions of them, but the error rate is high, 20%, up to 20%. And the long range, as I said, the actual sequencing is done by Illumina, but before that we have the 10x instrument that links those reads together. So there are obviously many different algorithms to work on this kind of data. For short reads, we have TARDIS, Daily, Lampi, Manta, Pindal, CNVnator. So Pindal and CNVnator are among the oldest ones. Daily and Lampi are probably the most popular. TARDIS is a tool that we have developed with Feridun Hürmüzdiari. Since it is written in red, obviously it's the best one. The long read algorithms, these are some of the examples. I'm pretty sure I'm missing about 20,000 more algorithms total in this slide. So we have the Smart SV, Corgi Sniffles. Sniffles was developed by Mike Schatz's group, Nano SV, and so on. And there are also some multi-platform algorithms, for example, HISA and multi-break SV. So it actually uses multiple sequencing technologies together, so long reads and short reads together. So they can also leverage the long read information coming from PacBio or Nanopore, and they can correct the small mistakes using Illumina reads, which are shorter but more reliable. The long-range algorithms, again, the most commonly used technology today for long-range sequencing is 10x, although there are others such as loop genomics. And these are some of the examples. So these are the linked reads are among the newest data types in this slide, so therefore there are less number of currently available algorithms. And obviously the one written in red is again ours and the best. So how can you find the structural variation using sequencing data? Makshat already went through this, but we basically have 4 different sequence signatures of structural variation. We can use the read pair analysis when we have paired-end data. So those blue bars on the top right, they are coming from the same molecule, which is around 500 base pairs for Illumina. And the reads are about 100, 150. So we know that the average size of that blue molecule is about 500, but when we align those reads to the reference genome, if we see them to be separated by about 5 kb, 10 kb, that's going to signal us that there is potentially a deletion here. And you can come up with different formulations for insertions, inversions, and translocations, and also mobile element insertions. So with the read depth analysis, we basically just align the reads, so we don't really care if it is paired-end or not in this type of analysis. So we just count the reads, and if we see a depletion of coverage in the reference genome, it's going to signal us a deletion. If there's an increase, it's going to signal us a duplication. So for the split read analysis, so we can do this with Illumina as well, but it will be more reliable with long reads because it's going to increase our mappability. So basically, we are looking for breaks in the alignment of the sequence itself. So here we are again showing the deletion. So that read is split into 2 parts when we align them to the reference genome. Or we can do local or de novo assembly. We take the reads, we assemble them with any assembler such as Abyss for Illumina or Canu for long reads, and then we compare those contiguous segments with the reference. So let's just go quickly with those algorithms. So with the short reads, we have 2 major challenges. One, we have some genome structure issues. The genome is repeated. There are lots of segmental duplications. About 5% of the genome is covered with segmental duplications, so these are larger than 1 kb. And about half of the genome is covered with common repeats. Since this also causes misalignments of our data when we have short reads, because an ALU is 300 base pairs, there are a million of them. Although they are not exactly the same, they are still very similar. And if our read rank is 100, you already have a million possible mapping locations. We also have the sequencing issues, so the reads are not perfect, although they are the most correct ones compared to the long reads. We still have some errors. We have some GC bias, especially if you are using PCR before the sequencing experiment. Right. So let's go through those signatures one by one very, very quickly. We can do the read depth analysis. So we basically compute the depth of coverage in sliding windows. So let's say this is our sample sequence data, and then we start mapping them to the reference. So this will take a while. Okay, so let's say the blue part of the sequence to test is deletion, and the red part is duplication, and the black part is just unique diploid part of a genome. So when you have that, you just calculate the genome-wide average, and then you do some GC correction because those reads are biased against high and low GC regions. And then after you calculate the average and the standard deviation, you look for parts where this deviates, if it is too many reads or too few reads. So then you just count those reads, and the blue part is going to signal you a deletion, because in this case there are no reads mapping. There could be still some reads mapping, especially if it's a heterozygous deletion. For the duplicated segments, you're going to see an increase in read depth. So these are some of the tools that only use read depth, such as CMNator and EWT. If you have exome data, it is actually messier. because now we also have to deal with the biases of the exome capture. But some algorithms that you can use are XHMM, Conifer, and exome CNV. So the read depth analysis assumes that it is a Gaussian distribution, or you Poisson distribution, but it is not. So as I said, it is affected by the GC content, for example. Although, again, you can get better data if you do PCR-free sequencing. And you can get very different distributions depending on the specific sequencing experiment you have done. So these 3+ are read depth distributions from 1000 Genomes project. As you can see, the leftmost one looks like the most Poisson-ish, although you still obviously have some noise on the left side, but the other ones are even dirtier. So when you do those read counts, you can, as I said, you can find duplications. So for example, this is from 5 different genomes. The red parts are the increased read depth regions, and the green parts are the normal read depth regions. And you can find copy number polymorphism. So for example, this part is not duplicated in the genome of Craig Venter, but it is duplicated in all the other genomes in this slide. And this part is not duplicated in the genome of NA12878, but it is duplicated in the mother's genome. So 12892 is the mother of this sample. It is duplicated there, but not duplicated in the father. So you can also try to differentiate the segmental duplications. As I said, they are very highly similar, by definition more than 90%. Some of them could be 100% exactly the same in the reference, but most of them have some differences. So you can also try to type which version of the duplicated gene is duplicated more or deleted, right? So for example, this is from Peter Sudmant from 2010. So this is an example of the amylase gene family where we have 6 copies in the reference genome, so 6 versions of amylase. So the amylase 2 are expressed in the pancreas, amylase 1 are expressed in saliva. This slide shows the predicted copy numbers of each paralog in the genomes of 180 individuals from 1000 genomes. So the idea is basically if you have those genes duplicated, you line them up. So this is the reference version on the top. And then you find the small differences between them. So for example, the G on the first sequence is specific to the copy 1 only, and the other copies don't have that G in that position. So if you see an increase or decrease of coverage of that base, then you can say that version of that paralog is duplicated or deleted. So here the black boxes are diploid, so that means you have the normal 2 copies of a paralog, and the blues and the reds are the increased copies. Here you cannot really see the top line is basically shows you which population it comes from. I believe the red ones are the Chinese population, the Asian populations, and you can see increased copy number of amylase 1B in Asian populations. And the deletions are going to look similar, so it will just be less reads aligning. So this is genomes of Craig Venter and an African genome. So you can see almost no reads mapping in the LCE3C gene in Craig Venter in Watson, sorry. So which corresponds to a homozygous deletion, or just a depletion in the coverage, but it's not 0, which signals you a hemizygous deletion in the African genome. So these were the first, the read depth was actually the first sequence signature used for CNV discovery, copy number variation discovery. And in 2005 and beyond, we started using read pair analysis. So the top left one signals the deletion, so this is the same example that I've shown before. And the other formulations basically show you the ways of discovering other forms of variation. So the top right is for mobile element insertion. So for example, that pink bar is a new insertion of a mobile element, and read pairs are coming from left and right of it. So the ones that are coming outside the mobile element are going to map close to each other in the reference, and their mates that are coming from inside are going to map somewhere else in the reference genome, which is going to be a representative of that mobile element type. Again, you can come up with inversion, translocation, and novel sequence insertion formulations. So one of the first algorithms that we have developed with Feridun 10 years ago now is variation hunter. So we were using the maximum parsimony approach. We were basically formulating the problem in set cover and approximating a solution. And there have been similar approaches developed, such as Hydra, Clever, Breakdancer, and then GASV. So these are all pretty old. These are the first read pair only algorithms. So the read pair also has some problems. So again, you assume a Gaussian distribution of the size of those short molecules. So this is a good data set. So as you can see, it is a pretty tight Gaussian distribution. And what you do is you plot this, calculate the average and the standard deviation of the sizes genome-wide, and then you flag those mappings with that map too close to each other as insertions and too far away from each other as deletions. But again, some distributions can be bad. Again, this is from 1,000 genomes. So the part of the distribution looks like Gaussian, but you can see the big tail on the left side. Since I don't really have 5 hours to go through these, I'll just show you the main ideas of read pair analysis. So again, let's say here we have an insertion in the bottom. That yellow box is inserted, and then from left and right we have the paired end reads. So we know the average size of this PE molecule. So the paired end molecule, again, for them it's about 500. And then we map them to the reference genome. If they are too close, it will signal an insertion. And it is also going to show you the approximate size of the insertion as well. So if the bottom is supposed to be 500, and if those reads are mapping within 300, it will likely show you an insertion of about 200. Right? So split read analysis. So you can use this obviously with long reads and short reads. So this is again similar. You are looking for breaks in alignment of each specific read. You can find deletions, mobile element insertions, inversions, and so on. Okay. So I'll just name a few. Again, these are split read only. So nobody actually does a split read only algorithm anymore for short reads. For long reads, we do. The first algorithm was pinned out by Kaye, and then there have been other developments such as Shrik and split read. So for PacBio and Nanopore, the splits are going to be much more reliable, especially if you have a mega-based long reads. And some examples are SmartSV that requires Blazer aligner, Sniffles by Mike Schatz, which requires or prefers NGMLR. And NanoSV, Swim, and Picky are some of those examples, and these are much more recent. So the last sequencing signature that you can use is assembly. You can do either de novo assembly. You just take all the reads and assemble a genome and then compare genome versus genome, or you can do local assembly if you are looking for insertions, for example. And the examples, algorithms for these are NovelSeq, Pamir, and Poppins. So basically, when you have this blue box is the novel insertion, which is longer than the paired end distribution, you are going to have on the left and right side of this insertion what we call one and anchored reads. So the ends that are coming outside the insertion are going to map to the reference genome, but the ones that are inside are not going to map. And the ones that are specifically entirely inside the insertion are not going to map at all. So we take those, we do a local assembly, and then we may merge those, the information coming from one and anchoreds and the information coming from the orphan reads together. So again, these are the examples, and I just heard yesterday that Poppins 2 is coming soon. So these are the single signature methods, but that work for some relatively simple structural variants, but there are more complex ones, and they are harder. So these are, this figure comes from the 1000 Genomes project. So basically these are only the calls reported as inversions by the 1000 Genomes project, but when you look them deeper, you are going to find that about 54% of all those inversions reported by 1000 Genomes project are in fact inverted duplications. So they are not inversions at all, they are duplications. Only 20% are really inversions as reported, and there are more complex events such as inversion and deletion, and multiple deletions and inversions going on in the same location. Okay. So even though you think that you can find inversions, actually you can only 20% of them are really inversions. So the reason is as follows. So if you only use the paired and information, for example, some of those events are going to show you the same kind of sequence signature. So for example, on the left-hand side, you can see the inversion and the inverted duplication. And if you are sequencing those gray arrows from the genome, and when you map them to the reference genome, it's going to look like those green ones on the top left. So the one that's coming outside is going to map on the reverse strand. The one coming inside is also going to map in the reverse strand and far away from it, which is exactly the same in inverted duplication. So in this case, we have those red arrows duplicated and inverted, right? So duplicated in inverted orientation. And if you have a paired and coming from the right part, it's going to map exactly the same as an inversion, which is also the same in the gene conversion event as well. So the same paired and mapping information is going to signal 3 different variants. So using only the read pair, you won't be able to separate those, which is the same story on the right-hand side. So deletions, interspersed duplications, and the direct orientation gene conversion are also going to give you the same paired and signal. So the solution, obvious solution, is using multiple signatures. So now you're going to integrate read depth, read pairs, novel assembly, and split reads, combinations of them, or maybe all of them together. So one of the first methods that came up with this was Lumpy by Ryan Layer in 2014. So which basically starts with the paired ensample and then refines using split read information, and then filters the deletions and duplication calls using the read depth information. More recently, again with Feridun, we have developed TARDIS, which also integrates split reads, read depth, and paired and information, although in a different way than Lumpy. As I said now, the most Illumina-only structure variant callers are now using multiple signatures. Genome strip uses read depth and read pair together. DELI uses read pair and split read together. SWABA uses read pair and assembly together. Lumpy, WAMP, TARDIS uses read pair, read depth, and split read together. Right? And MANTA and GRIDS uses read pair, split read, and assembly together. So you can see different combinations of sequence signatures. So these are multi-signature SV callers. So you just run Lumpy, for example, on a BAM file. Or you can use Ensemble algorithms to even get more refined calls. So one of the reasons is this slide. So this is again pretty old. This is from the first phase of the 1000 Genomes structural variants, where we used 19 different algorithms on the same data set to come up with a call set. Right? So some of them use paired and some of them use read depth and so on. And then what we did, actually Ryan Mills, who's in the University of Michigan, did this. He basically wrote a bunch of Python scripts to merge those call sets. So the structure variants, since they are coming from the complex regions of the genome, even if you are dealing with the same variant, even if it's a simple deletion, different colors may be off by a few base pairs from each other. So one of them says position A, the other one says position A plus 5, for example. Okay? So he came up with a bunch of scripts to merge those calls as reliably as it can, which could be regarded as the first ensemble algorithm, although it is pretty simple. So another reason is that no method is comprehensive. So again, this is from the same call set. Using 6 different read pair only algorithms, you can find a few thousand deletions, and these are all validated real deletions. And you can find less using read depth and split read, and their overlap is also very, very small. So the ensemble algorithms basically aim to combine the information coming from different callers. So there are different approaches. Intersection, for example, you just merge those that overlap, let's say more than 50%, more than 80%, or maybe you just take the intersection part, or you make a union of them. So that's what Ryan Mills did back in 2010. But obviously there are more involved algorithms that do this in a better way. Or you can use some validation information coming from other sources. Or you can use some other machine learning algorithms that you basically train with the known variants. So some of the tools for this are SpeedSeq, SV tools, HugeSeq, SV merge, and Parliament2 and Fusor SV. For exomes, there is a newer method called CNlearn from Pennsylvania State University from Santosh Girirajan's group. So this is the short read part. So again, you want to use multiple signatures and you want to use different colors. So Mike Schatz also said this, do not use just one color, use multiple of them, and then try to merge those calls using one of those ensemble algorithms. So with the long reads, the method that you can use is going to be the split read. So here is a difference between short reads and long reads. So let's say you have 3 copies of a repeat, those yellow boxes in the reference, and in the sample, the segment between repeat 1 and repeat 2 are deleted. When you have short reads coming from, one coming from repeat 1, one coming from repeat 2, they can map in different ways. So this maps here, or here, or here. So you have 3 different mappings, or this one maps here, here, and here. So 3 different mappings here. So in total, you have to deal with 9 different combinations of this pair band. But with the long reads, if the read is long enough to cover a repeat, what you'll do is basically you are going to use the parts that correspond to the unique segments to anchor them. Now you can separate the repeat 1 and repeat 2 from each other, and the split read information is going to show you where the deletion is. Okay? I'll just show a couple of examples. So I believe this is the exact same figure that Mike Schatz used 2 days ago. This is from their paper, which is mapping generated by NGMLR. And the variant is called using Sniffle. So you basically find this split. So on the left-hand side and right-hand side, they are the same reads, but their alignments are just split. And they are pretty much very much in agreement with each other. And these are some of the other algorithms. You can use long-range information, linked reads. So this is how it works. You take the DNA, you generate high molecular weight molecules. So these are on the average about 50 kb each. And then you dilute it. So this is all done automatically by this instrument from 10x Genomics. It is diluted into a number of pools, up to 4 million pools for 10x. And when you dilute this, there's a very low chance of overlap between them. So on the average, in each pool, you have 2 or 3 molecules, about 50 kb, and the genome is 3 billion, right? So their overlap is almost 0. And then you barcode them, and then you sequence using Illumina. So you again deal with short reads. But now you can track which read is coming from which pool, right? So those reads are, for example, coming from barcode 1 or pool 1. So if they are mapping kind of close to each other, they are coming from the same molecule. If they are very unrelated from different chromosomes, they are coming from different molecule. Same for the green one and the blue ones. So these are the properties. The average molecule size is 50 kb. You don't need any cloning. So the first method that came up with this linked read information used cloning back in 2011, but this is now automated process. But the size distribution is not tight. Per molecule, you have very shallow depth. Only 10% of the molecule is covered. But the total coverage is going to be 80, 90x genome-wide. So you can have up to 4 million pools. So statistically, you can have 20 molecules per pool, but in reality, you get 2 or 3 in real data. And these are some of the SV callers, and long ranger is the official caller from the 10x Genomics company, and Valor is ours. And there are others such as Grok SVS and Neighbor. So this is these methods use what we call a split molecule signature. So let's say we have an inversion. So this is the reference, and this green segment is inverted, right? And then you're going to have a bunch of molecules that are covering from those. So ideally, and it also works in reality as well, those grays, greens, and blues are coming from different barcodes. So they are barcoded differently. They are coming from different molecules. And you can separate that they are coming from different molecules. And these are coming from the genome that you are sequencing. And then you map them. So these are just going to map outside the non-inverted part. This is going to map inside the inverted part, but you're not going to actually see anything. And what you will see is it will be similar to the split read, but it will be in a much larger scale, which we call the split molecule. So these are going to be split from each other. Again, those reads are going to have the same barcode, right? So they are coming from the same molecule, but they are going to map a bit away from each other. And same story for the blue ones. Right? And additionally, you can also use read pair information, obviously, because they are Illumina at the end. So you do have paired end information. So for example, let's say these are coming from the inversion breakpoints, and they are going to show you the typical read pair signature for inversions. So now this is what we call this split molecules. So on the top, we have the inverted haplotype. So this is the non-animated version. You have the blue and the green molecules coming from different barcodes. And when you align them and then you look at the contiguity of the mapping, they are going to be broken. So you expect about 50 kb contiguity, but if you see 20 and 30 separated by 1 million base pairs, it may tell you that there is a 1 million base pair inversion in this location. And again, you can refine using the paired end information. So how do you identify the submolecules? As I said, these are shallow depth. These are coming from different barcodes. So basically, you just group those reads that are mapping close to each other and coming from the same barcode. So this you can group, and this you can group, and this you can group. That we call the submolecules. Right? So because you merge this information from the mapping. So these are two of the algorithms, Valor on the right, citation, and Neighbor on the left, but they are using the same strategy. And then you detect the structural variation using this split molecule information. Again, Neighbor and Valor are doing this a bit differently, although the identification is the same. Neighbor starts using the paired end information immediately with the split molecule information. With Valor and Valor version 2, we first try to find the potential candidate splits from the mapping information, and then we cluster them together. If they agree on something, we put them on a graph, and then we denote the agreement with the edges. And then we try to find the maximum clicks obviously, these are not going to do full clicks, but quasi-clicks, and we are going to find an approximate solution for it. And then we add the paired end information to refine and also filter the wrong calls. We also add read depth information to separate between inversions and inverted duplications, for example. And these are, some of the identified SV types. They are going to be similar to split reads, but we are going to formulate them as split molecules. So there are emerging technologies. I believe I have like 2-3 minutes left. So these, I'm just going to mention 2 of them. One of them is called Strand-Seq. So it requires a lot of lab work. So that's why it's really still emerging. So what you do is you basically start with those chromosomes, and then you label them with something called BRDU. I cannot even pronounce the full name of that thing. So basically what it does is you can mark the Watson strand and the Crick strand. And then when you apply, when you let those cells go through the mitosis, you are going to have some cells labeled and some cells not labeled, and then you can separate them. Basically, some cells are going to only have the Watson strand, some cells are going to only have the Crick strand. So now this will be single stranded cells, right? So you don't have Watson Crick strands in a cell. So it's either entirely Watson or entirely Crick. And then when you do single cell sequencing on those, so this is an additional source of noise in the data. If you're having a Watson only cells, you're going to see mapping information like this. If you have Crick only cells, you're going to have the mapping only on the positive strand. So Watson are negative strand, Crick are on the positive strand. So this will be the normal one. So what happens when there is an inversion? So in the reference, you have the negative strand and the positive strand, but in the inversion one, they are mixed. So some of the positive part is now moved to the negative part is moved to the positive. And then when you take them and then align them, this is what you're going to see. If there is nothing, you'll only see negative or only positive. But if there is a heterozygous inversion, you're going to see a lot of the negative and a little bit of positive, or vice versa, right? A lot of the positive and a little bit of the negative. If you have a homozygous inversion, you are going to see clear breaks. So negative, positive, negative. Or in some cells, positive, negative, positive. So since this is emerging, there is only one algorithm that deals with this kind of data, which is called BAIT. The last technology that I'll briefly mention is not sequencing at all. It's called optical mapping. So what you do is, again, you start with high molecule lower weight DNA, and then you use labels. So basically, you label those segments that are 6 or 8 base pairs long with different fluorescent molecules, and then you scan. Since you know which small motifs that you are labeling, you can also find their locations in the reference genome, right? So here. So here, for example, the green part is the supposed locations of those marks in the reference genome, and the blue boxes correspond to your sample, and then you just compare the locations and the distances between those motifs between the reference and the sample and the data that you get from the optical mapping. So for example, here you can find deletions because you are missing 2 of those marks, or you can see an additional mark in this case, and those marks are the other marks are coming too close to each other, which signals an insertion and so on. I am aware of 2 different algorithms that deal with this data, OIM-SV and Bionona-Solve. So further reading, so we already gave you about 5 papers to read about this talk, about this tutorial, but there are 2 newer ones that just came out very, very recently, a couple of weeks ago. There is a structure variation evaluation paper came out in Genome Biology. And there is a new version of Genome in a Bottle call set, which is available in bioRxiv as of June. So, in short, balanced rearrangements and complex SVRs are hard to detect. Short reads lengths make it harder, but it's better with long reads, better with linked reads, and also additional data coming from Strand-Seq or optical mapping helps, or even Hi-C, which I did not cover in this tutorial. And SNPs are boring. So this is something that Elaine Mardes said in the first 1,000 genomes meeting over 10 years ago. So I'd like to thank my group. Most of my students, almost all of them, have been at CGSI, and this time it is Zülala's turn. I'm guessing you already met her. And my collaborators and our funders, and of course, most importantly, I'd like to thank this grant. And you all forgot about this grant. So this is the CGSI grant. Where is Elazar? This is the most grant ever. Most important grant ever. All right. Thank you.